Hello, and welcome back to Human Evolution. Today we want to talk about classification. We also want to talk about anatomical evidence for evolution, but let's start talking about classification. And we've talked about this a little bit before on the first day when we were talking about one of the fundamental ways that we understand the world around us is to classify stuff, to put it into little groups. And so we need a system that's going to allow us to make sense of all of these very complex systems and the sheer number of things that we have to deal with, right? So the goal of any classification system in biology is to organize living things into groups that have a biological meaning, because it's going to make it easier to find information about that, easier to identify a new one when you see it, and it's going to show evolutionary relationships. So we do this in a couple of ways. The first thing that we're going to talk about two things. We're going to talk about taxonomy, and we're going to talk about systematics. Taxonomy is simply the science of naming organisms. We're going to identify them, we're going to arrange them into groups, classify them, and then we're going to give them names, right? And that's nomenclature. And the father of this system is, of course, Carolus Linnaeus, Carl von Linné. He liked his system so much that he actually gave himself binomial nomenclature. And what he said was that different species can be grouped into these more inclusive and more exclusive groups based on the characteristics that they shared. So a grouping of animals that is going to share a particular set of characteristics that's decided on pretty much by common agreement. Those groups are going to be called taxons, hence taxonomy. And so when we look at modern classification, right, we have 3 domains, and those domains are divided into kingdoms, and then we'll talk about the phylum, class, order, family, genus, species in just a second. But you're familiar with these different taxons, right? And so here's basically a breakdown, right, of what's in each one. And this is, of course, the taxon that we're interested in, the domain Eukarya. And it pretty much has all of the things that you really think about when you think about, if you picture any plant or animal, any organism, it's pretty much going to be in Eukarya, unless one of your things is bacteria, right? In which case you're dealing with these two domains. Okay. So when we talk about modern classification, we have all agreed that domain is the most inclusive classification, right? Those are the most inclusive taxa. And there are 3 of those. They're based on RNA differences, so that's how we classify domains. Then we divide those domains up into kingdoms. Then we design, and actually domain is a later addition onto the taxonomic system. It used to just start with kingdom, and so that's why we have that kind of interesting sort of double top layer of domain and kingdom, because domain was added later to the system. So just as a side note, but it's what we have now. So then we move down to kingdom. So if you look at ours, right, we start out in the domain Eukarya, and then we move into Animalia, then we move into our phylum, which is Chordata. These are things that have a spinal cord, not necessarily, or a notochord, not necessarily things that have a spinal column, right, but things that have a spinal cord. We are in a subphylum right here called vertebrata, which narrows that down to things that have a vertebral column. Then we can narrow that down even more to our class, which is Mammalia. You'll notice that the snake and the fish drop out right here, right, because they are not mammals, and so we're left with all of these guys. Then we can make that group more exclusive, right? And we can move down to primates, and that's where the elephants drop out. And then we can even go further and go family Hominidae, right? We're going to drop out gorillas. Then we can get even further. We can get down to our genus, which is the genus Homo. And we'll actually be differentiating the genus Homo from other genuses in the family Hominidae, and we'll talk about that more later on. But finally, we're going to get to our single species right down here, which is Homo sapiens. Now, an easy way to remember this is, does King Philip come over for great spaghetti? And that will help you remember the order of these things. Okay, so now when we talk about the species names are special. It's called binomial nomenclature. That is the scientific name, and all species are identified by both their genus and their species, and that is really important, okay? This is the scientific name. So, and this is really important because this animal is a cougar. This animal is a puma. This animal is a Florida panther. This animal is a mountain lion. This animal is a catamount. What do you notice about all 5 of these cats? I hope that you said that they're all the same cat, right? It turns out that what we think of as mountain lion or cougar actually has 5 different common names. So the common name is what everybody just sort of calls it locally, right? It would be very confusing to somebody if you walked up and said, "Ooh, I saw a cougar," and they were like, "Oh, I don't know what that is," right? Because I have mountain lions where I live. Well, a cougar and a mountain lion are the same, so we can unify and talk across local differences by having a scientific name. And in this case, it's Puma concolor, which is the cougar, puma, panther, mountain lion, or catamount. Okay, so this scientific name is unique to each type of organism regardless of its common name, right? And this goes the other way too. When I say robin, you probably picture this bird. But if you were in Europe, right, you would probably picture this bird. These are not the same bird, right? And so you can see they have different scientific names. You might also, in fact, picture this bird, right? These are all locally where they are called robins. And in fact, they are all different species. Does that make, I hope that makes sense. If you have any questions about this, please ask in class. Okay. All right. So talking about species, what is that? So which of the following definitions will describe species best? Similar individuals that live together, a group of similar individuals that interact and interbreed with one another to produce viable offspring, all plants, animals, and microbes, organisms that interact among themselves and their surroundings to maintain the ecosystem, or organisms that are recognizably the same. So ultimately, the basic definition of a species is a group of similar individuals that interact and interbreed, and this is the key, interbreed with one another to produce viable offspring. Viable offspring means that those babies can have babies, okay? And so this is what we're talking about in terms of species. You have to be genetically similar enough to have offspring. Now this goes to a second concept, and there's the biological concept of a species, and then there's the phylogenetic concept of the species. And so really when we define species, what we're going to do is we're going to balance that interbreeding to produce viable offspring with the genetic makeup of the organisms. We've run enough genomes now that we can start to see genetic similarities and differences. And I'll give you a really good example of this. Dogs and wolves. Number one, dogs are not recognizably the same. If you were an alien and you came to Earth and you saw a Chihuahua and a Great Dane, you would not think those are the same species, but genetically they are, okay? And so that's really important. That's where the genes come in. Now, having said that, dogs and wolves are still closely related enough that they can interbreed in the wild and produce viable offspring. And yet they are different species. Okay, because their genes are different enough and their phenotypes are different enough that we are going to classify them as different species. So there's some kind of flexibility in those definitions, but the standard is genetic similarities, looking for key markers, and producing viable offspring in the wild. Okay, so some of those traits that we use to classify species are also traits that we can use to look at evolutionary relationships. And so what we're talking about here is anatomical evidence, that things are related through form and function, right? These characters or phenotypes, right? These traits. So I just want to point out, when we are looking at traits and we're classifying and we're trying to group things, we can divide traits into 2 categories relative to any group. And those are the ancestral traits and the derived traits. Derived traits are traits that we use to classify a particular group, right? So for mammals, a derived feature is hair, because mammals are the only animals with hair, right? Now, for mammals, the presence of lungs is an ancestral trait, right? Because it's present in other groups. And you can take this a step further. So ancestral traits are things that other ancestors have, right? But they're not in that particular taxa. Okay. Now, if we go up, vertebrates, right? Lungs is actually not a characteristic of all vertebrates because fish have gills. And so you can get a sub, like, or you can get a super class, for instance, because you could drop fish out and you could classify reptiles, amphibians, mammals, and birds all together based on lungs. And if you did that, it would be a derived feature. Okay, so let me give you another example. So let's go with that hair idea, right? Hair is a derived feature in mammals because it characterizes mammals. We use that to decide this is mammal group. And this is like the activity that you did on the first day when you were trying to decide what traits you would use to group those groups into smaller groups until you got to the species level. Now, hair, for us, we are mammals, but if you look at the genus Homo, right? For the genus Homo, hair is an ancestral trait because it doesn't differentiate the genus Homo from any other mammal. So that would be an ancestral trait for us. So for mammals, hair is a derived trait. For humans, hair is an ancestral trait because other mammals have it. Okay? So when we talk about evidence for evolution, we're talking about these structures, right? So we're talking about which traits are ancestral, which traits are derived. We're looking for the similarities of two structures when we're trying to look at relatedness. We're looking at common ancestry versus environmental pressures. We also look at embryonic development, right? What do the embryos look like? What do they have in common? When are they changing? When are they developing? And then we look at vestigial structures, and those are things that have no function in one species, but you can see that they come from an older species, and I'll explain all of that in just a second. So let's talk about homology first. This is where we are looking at features that share common ancestry. And the best example, right, is the forelimb, right? So if you look at a pterodactyl, oh look, there's the forearm, there's the upper arm, there's the hand, there are all the little fingers, right? That's the little pinky finger, that's the little thumb, very exciting. Well, you can see this in a bird, right? All the bones are color-coded, by the way. Same structure. The fingers have changed a little bit, right? The fingers have changed. These have elongated, and some have been lost. That's okay, though, because you can still see the same bones, right? Same thing with the bat. Look, there's the little thumb. They're all four fingers. There's the forearm. There's the upper arm. So we're talking about the radius, the ulna, the femur. Okay, wow, no, sorry, humerus. Not femur. That's in your leg. Just kidding. It's the humerus. Humerus. Okay, so dolphin with its little fin, right? A seal with its little flipper. Guys, you can see they're all feet. They're all hands, right? Not feet, hands. They're all hands. So here's your humerus, radius, ulna, carpals, right? Fingers. This is us. This is our hand. But look, it's a hand and a shrew too. It's just that it looks like a paw. They all have different functions. You can see down here they all have different functions, but you can see that they all arise from common structures. Okay, so what's happened here is that over time, variation and the environment have allowed for these particular variations to be selected, and over time we've changed. And we'll talk about this more later on when we talk about natural selection. How does that work? But when we put it all together, you can see very clearly that these arise from a structure that is common in a common ancestor. It is called descent with modification, right? So ultimately what we're saying is that you have a group, but you'll have variety in that group, but you have a common ancestor that will have it. You can track it back to that common ancestor, and then everybody has some slightly different version of it. Okay. Now it turns out that within groups, we try to say, okay, let's not make phenotype the end-all be-all of classifying stuff, because you can get organisms that look really similar. Okay, a Patagonian hare, which is a rabbit, looks a lot like a guinea pig, which is not a rabbit. Okay, and they look very similar, but they are not very closely related. This is a very important thing. Having said that, though, oftentimes when you see something that looks similar, it probably is. Okay, so in other words, a cat's a cat. Now, organisms with a similar suite of features, right, what I'm saying is they do tend to be related. If you look at all of these guys, right, we've got two eyes in the front, we've got some sort of nasal feature, we've got a mouth, we've got two limbs on the front side, we've got two limbs in the back side, and it doesn't matter which one of these animals that you are talking about, okay? So they probably have a common ancestor. But when you start being more exclusive, right, if we're looking at, okay, what are some traits they don't share? Well, birds have feathers, mammals have hair. Okay, so we can drop that owl right out. Yeah. Grasping hands. Bears have paws, foxes have paws, but monkeys and humans have grasping hands. And so we can start to shift those into the same group, and we're all primates. Okay, so these similar traits really do help classify. So as a general rule, the more homologous characteristics are shared by two species, the more closely related they are. Okay, and we can use sequences of DNA and RNA and the proteins that those make as traits that we can use to look at common ancestry. All right, so these guys, even though they live in a completely different environment, this is a tropical environment, grassland, tropical grassland. This is a lion, right? This little fox, tiger, lion, related. Fox, fox, related. Yeah, because they share those suites of characteristics. Okay, now in an analogy, the characteristic does not share common ancestry. This is because of environmental pressures over time. And the panda's thumb is a really good example. Here is the panda's thumb. Here is your thumb, right? Now this is interesting. Let's count. 1, 2, 3, 4, 5. Wait a minute. All the panda's fingers are right here. So the panda's thumb actually isn't a thumb at all. It's an extension of the wrist bone. So if you bend your hand backwards, right, and you go to where your thumb is and you feel your wrist, there's a little bone there. Well, in pandas, that got really long and extended out. And now the pandas can use it a little bit like a thumb, but it isn't a thumb. And so this is a result of convergence. Homology is a result of divergence. This is a result of convergence. And so you can see, here's that extended wrist bone, here is your thumb. They are different. There's no common structure here. Common structure means common ancestry. Different structure means different ancestry. Okay? And so this goes to the idea of you can have similar features, right? The bat's wing and the bird's wing are homologous. They have the same common ancestry, right? But butterflies have wings too. They don't have bones. So this is an analogous trait. The wing of a butterfly is analogous because it does the same thing. Analogies do the same thing, but they don't have a common origin. Okay, so now let's talk a little bit about that embryonic development, right? So it's not just in the full-grown organism, the adult organism, or even during its development after it's born. We can look at before it's born and see similarities. You can see the fish, the chicken, and the human all have very significant similarities in their embryonic development. And so we can use that. So there are three big stages. You have your early stage, your middle stage, and then you have your late stage, right? And by this time, you've lost a lot of those similarities. But if you look at these guys, you can see that all of these vertebrates, they have common ancestry in their development. And that's kind of a cool thing. We're not going to talk about this a whole lot. If you are taking anatomy, we'll talk about this more. Okay, so let's talk about these vestigial traits, right? Vestigial traits are these throwback traits. So if you look at this dolphin, you can see that it only has 2 forelimbs, and yet one of the things that we said about mammals is that they have usually the number, limbs, right? So when you look at its little embryo, look, it has little cute legs, yay, right? But when you look at the adult dolphin, they're gone, yeah? Same thing with snakes, right? You lose structures that show up in development that indicate an earlier ancestry, some common ancestry of something, and as that is developed, right, as that organism develops, you lose those structures. Snakes end up having no little legs, okay? Now, this one is actually an adult. It's not an embryonic snake here, and we'll talk about what's going on here, but these are little, like, limb buds. So vestigial organisms are sort of remnants of past structures from other common ancestors. So this is interesting, though, because I just said that snakes lose all 4 of their limbs, dolphins lose 2 of them. So when a structure is present during development and then is lost, what's happening there? The cells dedifferentiate and redifferentiate into the cells required for new structures. The cells in the lost structures undergo apoptosis. They are consumed by macrophages. Remember, those are some of the not digestive, but those are the big cells that break down other cells that are produced by the organism's immune system, and they're released and float away just to dissolve someplace else. Well, it turns out that this is where we were talking about apoptosis, right? Those cells self-destruct and go away, right? So B is the answer to that. All right, so looking at some more vestigial traits, here is the vestigial pelvis in whales that indicates that its common ancestor at some point had a real pelvis and hind limbs, right? So it comes from a walking organism. Ostrich, right? Beautiful big wings. These actually are in a way vestigial because they are not used for flight. They're only used for courtship. Now, here's the thing about penguins, though. Do penguins have vestigial wings? Penguins do not have vestigial wings. Even though they don't fly through the air, they most certainly fly through the water, which is a fluid. And sometimes you lose them entirely. That is a kiwi. Kiwis have no wings at all. So sometimes traits will actually disappear over time entirely. Okay. Now, another thing about birds, they have hollow bones. Hollow bones minimize their weight, and so they don't have to use as much energy to fly. And in fact, they can fly. Penguins have hollow bones. The water supports the penguin really well, so the hollow bones are not something that is really beneficial to it, okay? But it still has them. It's a vestigial trait. Here is your little tail. That's vestigial, right? There it is, so tiny. Really annoying because it breaks really easily, though. So just as a side note, what about your appendix? You have this tiny little, mostly useless appendix. If you were a herbivore, like a squirrel or horse or something like that, you would have a much larger appendix. And actually, in the grazers, the grazing animals, the appendix is huge, right? It's used by herbivores. Ours is vestigial. Wisdom teeth, also vestigial. Our tiny little jaws really can't hold them very well, and we'll talk about these later on because they give us lots and lots of problems, okay? Look at those cute, fuzzy animals in the cold, all fluffed out because they're trying to get warm. You try and fluff out too, but you just don't have enough hair to make it matter. So later on, we'll talk about why it is that you are hairless, even though hair is a great coat. Yeah. Also, being bigger makes you look scarier. So that's why we fluff up, or try to, when we are nervous, afraid. That's why you get goosebumps when you're afraid. Okay. Muscular attachment in the ear. We can't turn our ears around like monkeys can because we don't have that muscle attachment anymore. And ours is folded over, vestigial. Okay. So what about nipples, right? Are nipples in men vestigial? They are not. They are just structures that we all have. It's just in females they develop into something useful. In males they do not. This is not a vestigial trait. So just because something isn't really useful doesn't make it vestigial. It has to be like a throwback. It has to be a remnant from something in the past. Nipples are very active for babies, right? Okay, so remember the kiwi? We talked about the kiwi that lost its wings. Well, can the kiwi ever get those wings back? Can structures that have been lost re-evolve independently? The answer is no. Once a structure is lost, it does not re-evolve from the same feature. Now, you may get another variation that allows you to get something that is a convergent trait, that's a similar trait, but it won't be the exact same thing that was before. So once a structure is lost, it cannot re-evolve independently. So this statement is False. Now, occasionally, your vestigial traits forget that they're vestigial, and you get an atavistic trait. That is when you get things like tails in people, right? See that little tail there? See that little tail there? That's not supposed to be there. An atavism is where the vestigial trait manifests more like the common ancestor. Okay, here are little legs in snakes. This is that skeleton that I showed you earlier that had the little snake legs. They end up looking something like this. Okay, remember the dolphins have legs when they are embryos. Occasionally a dolphin will get little leggy things when it's an adult, right? So given our common ancestry with other apes, right, what would be a good example of a possible atavistic trait that would show our close relationship? Would it be large canines? Would it be hooves? Would it be claws? Or would it be night vision? Well, chimpanzees don't have hooves. They don't have claws. They're daytime animals. But if you've ever seen a chimpanzee, and all monkeys and all apes actually have huge canines. So you could imagine we might have giant vampire teeth as an atavistic trait that shows our primate heritage. Okay, so now when we think about this anatomical evidence, this grouping by traits, right? We know that each taxon is characterized by a group of traits. We're going back to classification now. Each taxon becomes more and more exclusive. So in Eukarya, you got a bunch of stuff. Animalia drops out like all the plants and fungi. Chordata drops out everybody who doesn't have a spinal cord. Mammalia drops out everybody who doesn't have hair. Carnivora drops out everybody who's not a meat eater. Ursidae drops out everybody who's not a bear. Ursus drops out all the pandas and stuff. And then Ursus americanus is the American black bear that is only black bears. So we get more exclusive as we go down. Okay, and here's just another example. That's cats. Okay, so the taxon consisting of the most closely related species would be, in this group, the family, the genus, the order, or the class. Does King Philip come over for great spaghetti? It is the genus. And this works for plants too, okay? Here's just an example of corn. By the way, corn is the only thing in its genus and species. Oh, right? So we'll talk about that a little bit later on. But it's true for everything. It's not just animals, it's true for everything, right? So phenotypic and or molecular similarities are based on evolutionary relationships. True or false? This is actually false, right? Molecular similarities are based on evolutionary relationships. Some phenotypic similarities are based on evolutionary relationships, but some phenotypic similarities are based on convergent evolution, which would not have a molecular underlying foundation. Okay, so that is actually false. You want to be careful about that. Yes, for molecular similarities, maybe, maybe not for phenotypic. All right, so last thing we want to talk about is systematics. This is the classification by the phylogenetic relationships. Phylogeny is the line of evolutionary descent, right? How have things evolved? How are they related back? So when you look at the higher level of the taxon, the further back in time you're going. So when you talk about eukarya, you're going way, 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 way back in time to the common ancestor. When you talk about the genus Homo, it's way closer to the present. Okay, organisms that appear very similar, as we know, may not actually share a recent common ancestor, and so that's why this classification system is really important, because you are looking sort of at a chronology a little bit. So we talk about modern evolutionary classification. The original system was based on visible similarities, right? So a barnacle and a limpet, they both look the same. They're these little shell things. A crab definitely doesn't. It has legs, right? But when you start looking at evolution, right, and you start tracking those similarities, you can say, okay, well, they all have tiny little swimming larvae, right? But a limpet actually is a single piece. The organism is shaped a little bit differently. Segmentation and a molted external skeleton actually characterize both crab and barnacles, right? These guys will grow new shells, whereas limpets don't, yeah? And that's a very big difference. So even though they look the same, because they do the same thing, they sort of like stick to stuff, and they have these little shells, they're not the same evolutionarily speaking because of the traits that have developed along the way. Okay, so what we're really talking about is a phylogenetic tree. All right, so when we start looking at relationships, we want to start to be able to group them by traits. Okay, so here you have reptiles. Yeah, some dinosaurs are reptiles, maybe. We've really come to think, though, that dinosaurs are kind of their own thing, and so we want to reflect that properly. So what we're going to do is we're going to make a cladogram. Okay, so this is just a traditional evolutionary tree, but when we start looking at it by traits, the node here, this is where they connect, is going to indicate a common ancestor, and then you'll have traits. So there'll be some trait here that will separate mammals from turtles and everybody else. There'll be some trait here that'll separate turtles from lizards and snakes. There'll be some trait here that'll separate crocodiles from all these guys. Same thing here, okay? So, right, one thing you'll notice about a cladogram, which is a very special kind of phylogenetic tree, it shows trait relationships. It doesn't show time. This tree shows doesn't show time either. This tree doesn't really show good relationships, whereas a cladogram does. Okay, so a phylogenetic tree cladogram is going to show evolutionary relationships. We are going to do this in class. It does not indicate time or the sequence of evolution. Okay, so here's just a good example of a phylogenetic tree. Again, we're going to do this in class. So here you have vertebrates. Yay, all these guys are vertebrates. Bony skeleton. Well, we know that sharks are cartilaginous, and all the rest of these guys have bones. Oh, fish don't have forelimbs. All these guys do. Amniotic egg. We'll talk about that later on because that's really important for land stuff. Amphibians don't have that, right? Oh, hair. Oh, that's going to separate out just these guys. And eggs with shells. Primates and rodents and rabbits don't lay eggs, so that's going to separate these guys out. Okay, so do you see how that works? And when we put it all together realistically, we can start to get phylogenetic trees that do indicate age, right, and indicate evolutionary relationships, and that's kind of a nice thing. Okay, here's another one. The further back you go in this style of phylogenetic tree, the further back in time, right? So you're moving back in time here. Now, it's not always relative, but you can see that you have, it's not always chronological, it's not to scale, but you can see that as you go further back, you are going back in time. So remember, it doesn't give you the actual age, it just gives you the sort of like flow here. And that is not true on the cladogram. It's only true on the phylogenetic trees. Every phylogenetic tree is just a hypothesis. We've discussed that a little bit earlier. And then here's your last question. What happens when new data is discovered? The phylogenies change, phylogenies may not change, both or nothing. Well, as with all science, when new data is discovered, it might change something, it might not change something. So both of these things could happen. All right, so I know this was a little bit longer. We're going to build phylogenetic trees in class. We're going to look at it more. We're going to look at a little bit of anatomical evidence in class. But if you have any questions, please write them down and ask them when you get to class. All right, thanks for listening.